Welcome to part 2 of our nervous system. So this part will tackle more on electrical signals and neural pathway. So here we'll describe again the resting membrane potential, explain how the action potential is generated and propagated, and again compare the role of leak and gated ion channel. We'll also be describing the structure and functions of synapse and list the parts of the reflex arc and describe its functions. We'll also describe the converging and diverging circuit and the role of summations in the neural pathway. Okay, so let's start. So again, to review, this is our nervous system subdivided into central nervous system and peripheral nervous system. Central nervous system, you have your brain, spinal cord. Peripheral, you have the nerves, right? Cranial nerves and peripheral nerves. So in the cranial nerves, you have your neurons and the glial, right? Okay, so let's start with the organization of tissue. Okay, nervous tissue vary in color due to the locations and arrangement of the parts of the neurons and the glial cells. Again, the glials are the supporting cells, which does not undergo cellular division, right? Nervous tissue exists as a gray matter and white matter. Okay, so gray matter consists of group of neuron cell bodies and their dendrites, where there is a very little myelin. Okay. In the CNS, in the central nervous system, your gray matter on the surface of the brain, is called the cortex. So in the CNS, your cortex is the gray matter. Okay, it is in the CNS. So when I say cortex, that is your gray matter. Okay. And cluster of gray matter located deeper within the brain. So if it is located in deep part of the brain, we call that the nuclei. Okay. Similarly, the same with the PNS. PNS or peripheral nervous system also contain gray matter. Okay. But in the PNS, the cluster of neuron cell bodies is called ganglion. Okay. So in the PNS, this gray matter is called your ganglion, okay, consists of cellular bodies. Ganglia is from the word swelling or knot, buhol, right? Next is the white matter. White matter consists of bundle of parallel axons with their myelin sheaths with a white color. So this is your myelinated. White matter of the CNS form nerve tract. Okay, so white matter in the CNS, it has the nerve tract. Okay, nerve tract, or what we call the conduction pathway, which propagate action potential from one area of the CNS to another. While the white matter on the peripheral nervous system consists of bundles of axons. Okay, and it is associated with connective tissue that form the nerve. Okay, so in the PNS, we call these nerves or peripheral nerves. Okay, then let's review your membrane potential. It says that resting membrane potential RMP and action potential AP occurs in the neuron, okay? That is your cells. These potential are mainly due to difference in the concentration ion across the membrane, right? This membrane could be membrane channel and the sodium-potassium pump. The membrane channel include the leak channel and the gated channel. Leak channel and the gated channel. Leak channel are always open, you know that already. Whereas the gated channel, it is generally closed, but you can open it due to voltage. Either when it reaches the voltage, a certain level of voltage, it will open, right? That is your threshold voltage. Or if it is caused by chemicals or your neurotransmitter, gate is also will open. And you have two types of gated channel. Again, it's either the voltage gated channel and chemically gated channel. So leak channel are always open. Our ions can leak across the membrane down their concentration gradient because they are 50 to 100 times more potassium leak channel compared to sodium leak channel. So marami potassium, kunti lang ang sodium. Okay. The resting membrane has much greater permeability to potassium than sodium. Therefore, we know that the potassium leak have the greatest contribution on your RMP or resting membrane potential. Then the gated channel are closed always until opened by either your chemicals, your neurotransmitters, or the voltage reaching the threshold of your action potential. Okay? When open, the gated channel can change the membrane potential, thus the responsible for the action potential. Next is your sodium-potassium pump. Sodium-potassium pump compensate for the constant leakage of ion through leak channels. Sodium-potassium pump required to maintain greater concentration again, sodium outside, potassium inside. The pump actively transports potassium into the cells while sodium out of the cells. Okay. It is estimated that the sodium-potassium pump consumes 25% of cellular energy and 70% of energy in a neuron. Okay? 25% in a typical cell. So marami ang makukonsume ng neuron energy. Okay? So the resting membrane potential exists. Why? Again, the concentration of potassium being higher inside the cell and sodium outside. The presence of many negatively charged molecules like the protein inside the cell which attract the potassium, right? And we know that the opposite charge ions usually attract each other. The presence of leak protein channel in the membrane that are more permeable to potassium compared to sodium. Sodium tend to diffuse into the cell and potassium tend to diffuse out following your concentration gradient. So in order to maintain the resting membrane, again, they will use the pump, sodium-potassium pump. Okay? So the sodium inside the cell, which is much lesser compared outside, they will use the energy to go out, which is against the concentration gradient. The same with the potassium. Potassium that is outside the cell, in order to go inside the cell, again, they will use the pump. At the same time, they need energy to go inside the cell, which is more higher in concentration gradient. So this is the model of your resting membrane potential RMP. Okay, you can see that both potassium and sodium do have the leak channel, but it is the potassium that has a more numbers of gated channel of leak channel compared to sodium, right? It's the violet is your potassium and the pink is your sodium. There are more potassium leak channel than sodium leak channel. In the resting cell, only the leak channel are open. and the gated channel is closed. Okay, because of the ion concentration difference across the membrane, potassium diffuses out of the cell down to the concentration gradient. Okay, the tendency for potassium to diffuse out of the cell is opposed by the tendency of positively charged potassium to be attracted back into the cell by negative charge inside the cell. Okay, we know naman that inside is negative and positive is outside. So those potassium that is outside, they were attracted by this protein. This protein inside are negative. So when the positive charge, negative charge, they always tend to attract each other. So potassium again is pulled back to the inside of the cell. When the potassium came out, we know that it follows the concentration gradient, right? Potassium is higher inside, so it's very easy to go out to a much less concentrated area. So here, the sodium-potassium pump helps maintain, this is your sodium-potassium pump again. So it helps maintaining the differential level of sodium-potassium by pumping 3 sodium, okay, from inside going outside. So 3, right? And in exchange, 2 potassium from outside going inside. Okay, so you have 2. Okay, so you need 3 and 2. Okay, out of the cell is equal to movement of potassium in and out. So that also makes the membrane be in a resting state, right? So outside will become more positive and inside is again more negative with the help of your sodium potassium pump. Action potential allow conductivity along nerve or muscle membrane similar to electricity going along an electrical wire. So the channel responsible for the action potential is your voltage-gated sodium and potassium channel, which are closed during muscle rest or resting membrane potential. When a stimulus is applied to the nerve cell following neurotransmitter activation, so chemically gated channel, what will happen when it opens? Sodium channel open briefly and sodium rush in again. So sodium diffuse quickly into the cells. This movement of sodium, which is called the local current, okay, that is the local current, causes inside the cell membrane to become positive, and a change is called your depolarization. If depolarization is not strong, the sodium channel close again, and the local potential disappear without being conducted along the nerve cell membrane. Okay, so if this is your nerve cell membrane, so once it is polarized here and did not met the uh, threshold level, so it will go back to a resting membrane again. If this threshold depolarization causes voltage-gated sodium channel to open, generally at the axon helix. Okay, so if ever that the charge reaches the threshold, so depolarization will start, or action potential will start. Okay, and usually it happens in the axon hillock parts. Remember the part near on the cellular body. Okay, so this is your axon hillock part. Okay, the opening of this channel causes massive 600-fold increase in membrane permeability to sodium. So when that occurs, the more sodium came in, and the voltage-gated potassium channel also begin to open. So as more sodium enter the cell, which depolarization continued to a much faster pace, causing a brief reversal of charge. The inside the cell will become more positive and negative outside. The charge reversal causes sodium channel to close. So when that happens, and it becomes negative outside, positive inside, sodium channel will close, and definitely the sodium cannot enter the cell. During this time, more potassium channel are opening and potassium leave the cell, and repolarization will occur. Okay. At the end of repolarization, the charge of the cell membrane briefly become more negative than the resting membrane. We know that already. When it when it goes beyond repolarization stage, we call that hyperpolarization. Okay, action potential occur in all or none fashions, which means uh, the fact that if threshold is reached, action potential will occur. If there is no threshold, which is not reached by your uh, electrical charge, there will be no action potential. The sodium-potassium pump assists in restoring the resting membrane potential. Again, your sodium-potassium. So this is the diagram here. It shows that your resting membrane potential is in the level of 70, right? And the dotted line is your threshold membrane potential. We say that the polarization start or action potential activated when the charge reaches the thresholds. So this is your polarization, which uh, stands for sodium influx, right? I hope you still remember sodium influx. And you have the repolarization here. Here, you know that potassium begins to leave the cells, right? It is your potassium efflux, right? And during repolarization, sometimes when the potassium went out of the cell, it goes beyond the resting membrane potential level. We call that hyperpolarizations. So this model shows your action potential in the nerve, okay? So you can see here, this is the neuron cell, right? And this sample is taken along the axon of your neuron. So this is during resting membrane potential, the sodium channel, which is the color pink, and you can see that the gated channel of sodium and potassium is distributed along the membrane of your nerve. Okay, so the outside of the cellular membrane, of course, is positively charged, and the negative is inside. Again, it's being controlled by your sodium and potassium. Gated channels. No? Okay. This is what happening during the depolarization. Your local potential, of course, right, reach when the sodium rush in the cells, and when it reaches the threshold, right, it will start the action potential or the depolarization state. Okay, it is due to the sodium influx. Then when it reaches the certain level, the potassium gated channel also will open, right? So that the potassium can leave the cells. Okay, so this is the continuation, right? You can see that now the graphic is pointing on the repolarization state. So when the peak period of depolarization reaches, the sodium gated channel will close. Sodium cannot get in, right? So no more sodium. but at the same time, your potassium gated channel open and potassium went out the cell. So more potassium will go out, right? Making the inside the cell again negative and positive outside the cell, outside the cells. So we say that sometime when potassium leave the cells, it's go beyond your resting membrane potential level. We call that hyperpolarizations. Okay. So next is the unmyelinated and myelinated axon action potential. Action potential are conducted slowly. We say that in unmyelinated, which means no myelin sheath, and more rapidly in those axons with covering or with myelin sheath. Action potential along unmyelinated axon occur along the entire membrane. Okay. Action potential on myelinated axon occur in a jumping pattern. Jumping pattern. We call that saltatory action. Saltatory action or saltatory conductions. So it's like a grasshopper that jumps from one node of ring beer to another node of ring beer. Okay, so this is the model of your unmyelinated axon conduction, which shows why it is slow compared to myelinated. Okay, so the action potential conduction will travel from cellular body going to the axon, right? Okay. So an action potential is the orange part of the membrane. Okay, generate local current that depolarize the membrane immediately adjacent to the action potential. So if this is the action potential, so it will generate slowly along its membrane. Okay, so when depolarization ceased by the local current reaches threshold, a new action potential produced adjacent to where the original action potential is occurred. Okay, you can see. So from here, it will go to the next part only after reaching the threshold, right? This one. Once it reaches the threshold, it can depolarize the adjacent part. That's why it takes long or slow conductions. Comparing to your myelinated axon conduction, okay? So action potential at a node of brain bear generate local currents, okay? The local currents flow to the next node of brain because the myelin sheath of the Schwann cell insulate the axon of the internode, okay? Since there is a covering here, it cannot travel. So instead, it will jump, right? So when the polarization caused by the local current reaches the threshold of the next node of ring beer, a new action potential is produced. So you can see here, right? This is the first. This is your local current. When it reaches the threshold, right? So it generates along this span. And the next part is this one. This is your node of ring beer. After generating here your action potential, it will jump again to the next node of beer. So action potential propagation is much rapid in myelinated axons because the action potential are produced at successive nodes instead at every part of the membrane along. The same what happening in unmyelinated axons. So next, axon conduction speed. So the speed of action potential conduction vary widely even among myelinated axon, which is based on the diameter of the axon fibers. Depende sa laki. Medium diameter, which is lightly myelinated axons, Characteristics of autonomic neurons conduct action potential at a rate about 3 to 15 meters per second. While the large diameter, malaki, heavily myelinated, maraming myelin sheath, conduct action potential at a rate of 15 to 120 milliseconds. So it is much faster compared to medium diameter. Next, the synapse. Synapse is a neuronal, which means two nerve, right? Remember the myoneural, which means from neuron to muscle. This one, neuronal synapse, which means nerve to nerve, okay? It is the nerve-to-nerve junction where the axon to one neuron interact with another neuron. The end of the axon form a presynaptic terminal, and the membrane of the next neuron form the postsynaptic membrane, okay, with a synaptic cleft between the two membranes. Usually the postsynaptic membrane is the dendrite part. Chemical substances called the neurotransmitters. Again, they were stored at the synaptic vesicles in your presynaptic terminal. So it's the same with your uh, skeletal muscle. An action potential reaching the presynaptic terminal again will causes the voltage gated channel calcium to open, and once it is open, calcium again moves into the cell. The influx of calcium will causes the release of neurotransmitter by a process of exocytosis. The neurotransmitter diffuse across the synaptic cleft, which is going to bind to specific receptors molecule which is located in the postsynaptic membrane. Next is the synapse. The binding of neurotransmitter to this membrane receptor, what will happen? It's either open or close the postsynaptic membrane, right? So the gated channel can be gated channel for sodium, potassium, or even chloride. The specific channel type and whether or not the channel open or closed depend on the type of the neurotransmitter, which is located in the vesicles of presynaptic terminal and the type of receptors on the postsynaptic membrane. So the response may be either stimulate or inhibit, increase or stop, okay, of an action potential located in the postsynaptic cells. If sodium channel open, okay, the postsynaptic cell become depolarized. You know that will happen. Once it depolarizes and reach the threshold, it will cause action potential, right? If potassium or chloride channel open, if it is the one that is located in the postsynaptic membrane, then the potassium will leave the cell, right? And the inside the cell will become more negative, which will lead to hyperpolarization, right? When that happens, no action potential will happen because action potential happen only if the reversal of charge occur, right? Negative outside and more positive outside. There are many neurotransmitters with the best known being acetylcholine and your norepinephrine. And you know already the acetylcholine. Let's the norepinephrine will be learning also what is norepinephrine. So neurotransmitters do not normally remain in the cleft indefinitely or forever, right? Thus, their effect will be short only in durations. Like for example, your acetylcholine will be breakdown for your acetylcholinesterase. So that can limit the polarization or the initiation of action potential, which can lead to contractions if it is in the muscle. Okay? So these substances become reduced in concentration when they are either broken down by enzyme or sometimes like your norepinephrine, it is being transported back into the presynaptic terminal, but can be also broken down by enzyme. Okay. Your acetylcholine, it's not transported back. Definitely, they will break it down. But your norepi can be sent back to the presynaptic terminal. Okay. So this is the model of your synapse. Okay. Synapse of your nerve cell to another nerve cell. So this is the axon, this is your presynaptic terminal, and this is your postsynaptic membrane. And of course, this is your cleft, right? Space in between is your cleft. And you can see the green receptor there, that is for calcium. Action potential lead that to open when certain voltage is reached, right? The threshold. When the calcium reach inside the cell, the vesicles will open and send those neurotransmitter, which is located inside the vesicles, outside the cleft. Okay, that neurotransmitter will attach to the receptor site in the postsynaptic membrane, which is uh, classified as chemically gated channel. So when it is open, for example, if it is for sodium, then sodium rush in, right? If it is for potassium, then potassium will go out the cell and there will be no depolarization that occur. But if it is for sodium, sodium will go inside and there will be depolarizations. There's a lot of neurotransmitters. It's not only acetylcholine and norepinephrine, right? Another neurotransmitter which you can encounter on your study is like your serotonin. Serotonin is also a neurotransmitter that you know already that it is your a happy hormone, right? It can influence your mood, your dopamine, your gamma or aminobutyric acid, okay, your GABA, short, your glycine and endorphins. Those were endogenous morphines. Okay, neurotransmitter do not normally remain in the synaptic cleft. You know that it can either broken down by an enzyme, your acetylcholinesterase. After breaking it down, it will send it back to the presynaptic for reuse purposes. Okay, so norepinephrine is either actively transported back without breaking. It can be sent back again to the presynaptic without breaking. Or it can be break also, the same with your acetylcholine. Okay. So the release and breakdown or removal of neurotransmitter occur so rapidly that a postsynaptic cell can be stimulated many times a second. That's why they're breaking it down so that no more depolarization can take place. Okay. There is certain drug. A drug can modulate the action of neurotransmitter in the synapse. For example, is your cocaine. Okay. This is a controlled drug. Cocaine and your amphetamines. Okay. Cocaine and amphetamines increase the release and block the reuptake of norepinephrine. So if there's no reuptake of your norepinephrine, meaning norepinephrine will stand long time in the cleft, right? Which result in overstimulation of the postsynaptic neurons and deleterious effect on the body. Okay, so drug that blocks serotonin, for example, serotonin reuptake, because usually your serotonin, those unused serotonin should be return it back to the presynaptic terminal. Okay, so there's also drug that can block the reuptake, making serotonin more available in the cleft. When that happens, particularly
it can treat the depression because we say that serotonin is a happy hormone, right? So the more serotonin can improve the mood and it can solve the depression state, right? It can improve the mood. Next is the reflex. Reflex is an involuntary action in response to stimulus applied to the periphery and transmitted to the CNS. Reflex allow the person to react to stimuli more quickly. It's possible if conscious thought is involved. Most reflexes occur in the spinal cord or in the brainstem rather than in the higher brain center. We have this reflex arc, which is located in the neuronal pathway by which a reflex occurs, and it has a five basic components. The five basic components of your reflex arc are the sensory receptor, the neurons, sensory neuron. Inside the spinal spine, you have your interneurons in your spinal cord, where the neurons are located between communicating to other neurons. The motor neuron and the effector organs. Effector organs can be a muscle or gland. Okay? So the simplest reflex arc do not involve the interneuron. The interneuron is located again in the spinal cord. So here is our model, the reflex arc. Okay, has five components, right? The sensory receptor, the sensory neuron, and you have the ganglion there, which is composed of cellular bodies, right? You have the interneuron located in the cord where it synapses to the motor neuron. So this is your sensory neuron and it synapses to the interneuron. Interneuron synapse in the motor neuron going back to the effector's organ, okay, via your nerve. Call that nerve. It is in the periphery. Then you have your skeletal muscle as the effector organ. So what is happening here? So when there is stimuli, for example, an injury in the skin, that sensory or stimulus will be carried by your sensory neuron going to the spinal cord. Okay, that sensory will be synapsed to the interneuron. So instead of going to the brain, it will synapse to a motor neuron. Okay, so the motor neuron already carried an impulse or the reflex that immediately respond to the effector organ without reaching the brain. You have already the response. That's why it is called a reflex arc. So reflex arc is the simplest or the smallest, the basic functional unit of the nervous system. Why? Because it's the smallest, simplest pathway capable of receiving stimulus and yielding a response. Okay? Okay, so next is the neuronal pathway. First type is converging. Sa converging pathway, it is a simple pathway in which two or more neuron synapse into one neuron. Okay, so two neuron connect to one. This allow information transmitted into more than one neuronal pathway to converge into a single pathway. Next is diverging pathway. It is a simple pathway in which an axon of one neuron divides and synapse with one uh, or more than other postsynaptic neurons. So meaning there's a neuron having multiple axon, right? So if it has more axon, it can synapse to one or more postsynaptic neuron, right? So from one can synapse into two. We call that your diverging. Okay? So this allow information transmitted in one neuronal pathway to diverge into two or more pathway. So like this is the model of your converging, right? This is your converging and this is your diverging. Okay, see, two neurons synapse into one, right? While this one neuron synapse into two. It has two axons here. So meaning the messages can be sent into two area. This one, messages from different area can be brought into one area. Next is the summation. A single presynaptic actions potential. So next is the summation and spatial summations. Within the CNS and in many PNS synapses, it takes more than single action potential to have an effect. So one single potential, sometimes it will not have an effect. It needs more action potential to have an effect. A single presynaptic action potential usually does not cause a sufficiently large postsynaptic local potential, which can reach the threshold and producing action potential in the target cells. So instead, many presynaptic action potential are needed, and that process is called summations. Okay, that process is called summation. So summations of signals in neuronal pathway allow integration of multiple subthreshold local potentials. Summation of the local potential can bring the membrane potential to threshold and trigger action potential. Okay, next is your spatial summation. It occurs when the local potential originate from different locations of the postsynaptic neuron. Okay, so this is an example of your converging pathway. It originate from different location, right? But it synapse in one neuron, which converges one pathway. So temporal summation occur when local potential overlap in time. Okay, so this can occur from a single input that fires rapidly and allow the resulting local potential to overlap briefly. Your spatial and temporal summation can lead to either stimulations or inhibition or stop, which is depend on the type of the signal. This is the end of part 2. See you on the next part, the part 3. Okay?